My name is Joshua Mugabi. Welcome you to topic 4, respiration. Our learning outcome B. Analyze the biochemical processes leading to ATP production in living organisms and how these processes are affected by physical activities and respiratory poisons like cyanide. Details of biochemistry are known to required. We have the learning activity C. In groups, learners search for the citric acid cycle, focusing on key steps: substrate level phosphorylation, decarboxylation, and the production of NADH and FADH₂. Groups then engage in a hands-on activity such as constructing a flowchart to visualize the cycle and present it in class. Citric acid, the Krebs cycle, item 1. Entry of acetyl-CoA into the cycle. After glycolysis, the formation of acetyl-CoA, respiration continues in the mitochondrion. Task: state where the citric acid cycle occurs. Explain how acetyl-CoA enters the cycle. Name the first compound formed. State where the citric acid cycle occurs. The citric acid cycle occurs in the mitochondrial matrix. Explain how acetyl-CoA enters the cycle. Acetyl-CoA, a 2-carbon, combines with oxaloacetate, a 4-carbon. Name the first compound formed. A 6-carbon compound, citrate, citric acid, is formed. The citric acid Krebs cycle, item 2. Let's now focus on decarboxylation steps. Carbon dioxide is released during the citric acid cycle. Task: define decarboxylation. State when decarboxylation occurs in the cycle. Explain the significance of decarboxylation. Define decarboxylation. Decarboxylation is the removal of carbon dioxide from an organic compound. State when decarboxylation occurs in the cycle. occurs when a 6-carbon and a 5-carbon intermediates are converted to 5-carbon and 4-carbon compounds respectively. Explain the significance of decarboxylation. It leads to complete oxidation of acetyl-CoA. Carbon is released as carbon dioxide. Energy is conserved in the reduced coenzymes. The citric acid or Krebs cycle, item 3. Let's focus now on production of NADH and FADH₂. A student observes that reduced coenzymes are formed in the cycle. Task: state the role of oxidized NAD and FAD in citric acid cycle. Name the reduced forms produced. State the importance of the reduced coenzymes. State the role of oxidized NAD and FAD in the citric acid cycle. NAD and FAD act as hydrogen and electron acceptors. Name the reduced form produced. NAD is reduced to NADH. FAD is reduced to FAD2H. State the importance of these reduced coenzymes. NADH and FADH₂ carry electrons to the electron transport chain. They are essential for ATP production by oxidative phosphorylation. The citric acid Krebs cycle, item 4. Let's look at substrate level phosphorylation. Some ATP is produced directly during the citric acid cycle. Task: define substrate level phosphorylation. State when it occurs in the citric acid cycle. State the amount of ATP produced per cycle. Define substrate level phosphorylation. Substrate level phosphorylation is the direct formation of ATP from ADP using energy from a chemical reaction. State when it occurs in the citric acid cycle. It occurs during the conversion of succinyl-CoA to succinate. State the amount of ATP produced per cycle. 1 ATP or GTP is produced per cycle. A citric acid Krebs cycle item 5. We are now looking at the summary of energy yield. A teacher asks learners to summarize the products of 1 ton of the cycle. Task: state the products of 1 complete citric acid cycle. Explain why the cycle must turn twice per glucose molecule. State the products of 1 complete citric acid cycle. From 1 acetyl-CoA, we get 2 molecules of carbon dioxide, 3 molecules of NADH, 1 molecule of FADH₂, and 1 ATP or GTP. Explain why the cycle must turn twice per glucose molecule. 1 glucose molecule produces 2 acetyl-CoA molecules. Therefore, the cycle occurs twice per glucose molecule. Citric acid Krebs cycle item 6. We want to look at the significance of the citric acid cycle. A cell's respiration rate decreases when enzymes of the citric acid cycle are inhibited. Task: state the significance of the citric acid cycle. Give 1 consequence of inhibiting the cycle. State the significance of the citric acid cycle. Completes oxidation of acetyl-CoA. Produces reduced coenzymes for ATP synthesis. Supplies intermediates for biosynthesis. Give 1 consequence of inhibiting the cycle. Reduced ATP production, accumulation of metabolic intermediates, cell energy deficiency. Marking emphasis. Correctly locate the citric acid cycle. Describe key steps in correct sequence. Clearly explain decarboxylation. Accurately state production of reduced NAD and reduced FAD. State ATP yield correctly. Conclusion. The citric acid cycle is a series of enzyme-controlled reactions occurring in the mitochondrial matrix. Acetyl-CoA is completely oxidized through decarboxylation, producing reduced coenzyme NADH and FADH₂, and ATP by substrate-level phosphorylation, which are essential for cellular energy production. Thank you very much for participating in topic 4, respiration. We have been looking at learning outcome B. Analyze the biochemical processes leading to ATP production in living organisms and how these processes are affected by physical activities and respiratory poisons like cyanide. Details of biochemistry are not required. We have been looking at learning activity C in groups. Learners search for the citric acid cycle, focusing on the key steps: substrate level phosphorylation, decarboxylation, and production of reduced NAD and FAD. Groups then engage in a hands-on activity such as constructing a flowchart to visualize the cycle and present it in class. If this has been useful, visit me at Mugabi Joshua Biology on YouTube.